throughout my career in drug discovery, I've tried to keep one foot on the basic research side and one foot in the applied or translational side. And so for many years, I've had a joint appointment at Yale Medical School, also at Woods Hole Marine Biological Laboratory, which is sort of the germinal layer for many neuroscientists throughout history. I also was a founder of a small medical devices company aimed at epilepsy in the Boston area, Therostat, and a company in London, which some fellows at the Imperial College and I have founded, called GlioQuell, which is going after glioblastoma. So still keep my hand in on both sides, although even though I'm getting a little long in tooth. Let's see, let me figure out how to advance these. Okay. So what I hope to discuss today, if I can move this one bar out of the way, there we go, is briefly what constitutes the central nervous system and why is CNS drug discovery so difficult. I'll also discuss categories of targets for drug discovery, types of diseases and conditions, what's happened to CNS drug discovery programs over the last 2 decades, some notable failures and successes, and what's changed or is changing that should increase interest in CNS targets. And I understand that I will go ahead and give a talk, and then there'll be some time for questions at the end. But if there's something that's absolutely pressing, feel free to bounce in. I originally gave this talk. I should give it a little history of how I was invited to write a paper with, and I invited one of my colleagues at Yale, Len Kaczmarek, who's a very famous ion channel guy, and that's where I do most of my basic research, is in looking at ion channels. My group was responsible for many ion channel cloning advances and finding modulators for those, and trying to advance the understanding of ion channels, particularly voltage-gated ion channels, as drug targets in the central nervous system. And originally, I spent almost 20 years at Bristol Myers Squibb, where I was eventually in charge of the CNS drug discovery effort on ion channels. And as a product of that, we developed a clinical candidate for stroke. And I will talk actually quite a bit about stroke as a target in this talk. But drug discovery and development, I don't know the backgrounds of all of the people on here, but I assume that you have either a professional interest or personal interest in drug discovery and development. It's a very difficult process.
Text them again.
Sorry? Oh. It's among the most heavily regulated industries. And I said that once at a talk, and somebody who worked originally as an aircraft engineer said, no, you're not. The aircraft are even more heavily regulated. And my comeback to that was, yes, but there's rarely been an aircraft that was designed that failed to eventually get onto the market, whereas most drugs are never approved. And either they fail because of efficacy or because of side effects, or a whole panoply of reasons that they don't succeed. Between 2010 and 2019, the average was about 35 new drugs per year approved by the FDA. Surprisingly, given the title of the talk, a fair number of these are CNS drugs. But if you look closely at it, many of them, most of them, are really Me Too drugs. And most of them are in the psychiatry area, a completely different subject that I've recently written a review about, but another very difficult area. So failure rates for drug candidates preclinical stage is greater than 99%. And that was described to me when I first started in the pharmaceutical industry. Somebody turned to me and said, this is the only industry on earth where you can be right almost 100% of the time, saying no to the advancement of a candidate into the clinic or for further development, and look like a genius, because almost everything fails. So if you thumbs down it, you're going to end up saving us a lot of money. Of course, we'll never have any drugs, and we won't do what we're supposed to do. But that's the way a lot of people work in the industry, and it makes it very, very difficult. There's a lot of politics involved. From phase one, once a drug enters the clinic, greater than 90%, and you're buying down risk of failure, obviously, as you advance through the clinic. But you know, in any discipline, very few drugs make it through the process of the initial discovery, translation into a clinical candidate, and then eventually the clinical study of that drug. This varies, of course, by the discipline, but CNS drugs tend to fail at a higher rate than most others, and we'll talk about why that is a bit. Drug discovery and development is very expensive. You know, the most frequently cited study is that of Tufts University, which has an entire area department that's sort of dedicated to looking at the economics of this. They estimate greater than 1 billion and greater than 12 years to market on an average. I think that's a bit overblown because they use industry figures, and you know, the industry, obviously it's in their best interest to make it look more expensive, at least in the United States, where they can then charge more money. But it's very expensive and you know, prone to great failure. It's a very difficult area to be in. The CNS is a particularly difficult organ system. Many CNS drugs, again, are approved, but most of them are Me Too drugs, and a lot of them are psychiatric drugs. There's been little to no progress in a number of areas. So as a drug target, and I'm going to be going through this quickly, but if there's anything anybody doesn't either follow or understand or agree with, you can always chime in. The ultimate targets are usually neurons and glia. Those are the primary cells within the central nervous system. Central nervous system is brain and spinal cord. There are a lot of peripheral nervous elements, but you know, those are really approached differently and separately oftentimes. There are billions of neurons. There are many different types, and within the central nervous system, there are numerous functional regional differences. And the thing that many people don't understand with relation to the central nervous system is that while there are many different transmitter systems with specific receptors or hormonal systems, voltage gated, ligand gated ion channels, ion pumps, and enzymes, and what have you, there's an enormous overlap between those. And I'll give you an example. Many people have targeted the excitatory amino acid system for things like new psychiatric drugs, for example. The problem is that glutamate, glutamic acid, which is a excitatory transmitter for fast excitatory transmission in the CNS, is used by about 95% of the known excitatory synapses. So if there's a problem in one particular area and you develop a drug that affects glutamic acid neurotransmission in that area, it is also going to affect it in every other area where there may not be an effect. And so that overlap of these systems, there are very few places where there are unique transmitter systems that are unique to a particular modality of function within the CNS. Dopamine is sort of one of them. Acetylcholine was thought to be for cognition, but that's not really true in general. The complexity of the biology means that diseases generally affect many different systems within the CNS. And similar disease phenotypes can result from very different underlying pathologies. You know, people lump Alzheimer's disease, you know, anybody who becomes demented, you know, cognitively impaired as they get older, they suspect or actually diagnose as Alzheimer's disease. Not necessarily the case. Obviously, there are many different types of things that can lead to dementia. That has been part of the problem, I think, with Alzheimer's disease, although that isn't the major contributor to the failures. Neurons, for the most part, don't regenerate. There's a period of development, but beyond that, neurons are not going to readily regenerate in most areas. function that therefore is already lost to a disease condition or some sort of other pathology, some trauma, may not be reattained. Even if you have a successful drug treatment, you may be able to stop further damage, but you're unlikely to easily be able to regain function. It is notoriously and truly difficult to get drugs into the brain. We've made a lot of advances in our understanding of how to do that, but it does involve some compromise with respect to the characteristics of small molecule drugs that are developed for brain penetration and can affect downstream how those drugs get studied in humans. And I'll give an example of that. Behavior is easily affected by drugs that enter the CNS. Everything about central nervous system function is behavioral. That's how we see whether something is right or wrong with the CNS. Phenotype really is behavior, and just about everything can affect behavior. So side effects are a huge issue with CNS drugs. I won't go into great detail on the blood-brain barrier, but the blood-brain barrier is formed by capillary epithelium in most brain regions and is designed by evolution to exclude things that shouldn't get into the CNS. I mean, that should tell you something if you really think about things in evolutionary terms about how sensitive the CNS is to any kind of perturbation, because there's an entire system that's evolved to keep things from coming in and perturbing that system. Specific transmitter systems also exist to exclude certain types of lipophilic compounds and toxins. In other words, if they get in, it spits them, or start to get in, it spits them right out. And there are also transporter systems that will specifically move things into the CNS. That blood-brain barrier excludes nearly all large molecule therapeutics and greater than 95% of small molecule drugs, unless they're specifically modified and designed to get across there. That's only recently started to be successful with large molecules. For example, peptide therapeutics. Traditionally you couldn't get peptides into the CNS, for love or money. Now we have designed ways, or I should say that medicinal chemists have designed ways that increase the probability that you can get them in and that they won't be torn up once they get in. It is a significant barrier to the development of CNS drugs. Not so much that we eventually can't find a molecule that can get in, but that many compromises are made in the structure of those molecules in order to get them in. And often, at least with small molecules, that means that highly lipophilic compounds are used to circumvent the blood-brain barrier. And it often means that those things have great solubility issues. And so it's very, very difficult to find an ideal candidate to get across the brain. Obviously, it can be done. There are many drugs that do enter the brain, but it's much more difficult than getting into other compartments within the body. Different barriers determine entry into and out of the cerebrospinal fluid and the retina. Some people look at cerebrospinal fluid as an indicator of brain entry, and that is not necessarily the case. Sometimes it correlates, often it does not. In terms of diseases and conditions that affect CNS, just about anything that you can think of. Genetic and developmental disorders are a large and growing number of things that affect the CNS through even single point mutations, things like Fragile X, the mutation of the mental retardation protein, and Tourette's and seizures disorders of infancy, which is one that I work on, one of the channels that I first cloned and found modulators of. The thing called the SLAC channel, or the sodium dependent potassium channel, is mutations in that channel have recently been found to be the genesis of migrating partial seizures of infancy, and a very terrible disease, rare thankfully, but terrible, and have led now to entire programs and a couple of companies that are pursuing it. Autism, which has many genetic underpinnings. Many other CNS disorders, including Alzheimer's and schizophrenia and ALS, there are at least a cohort of patients which are known to have a mutation which leads to the disease. And that actually has often led, I think, to a mistake in assuming that the large majority of the patients that are spontaneous with respect to the genesis of the disease have no known genetic linkage. That the same thing that applies to the small cohort that is genetically linked would apply to them. Yeah, and we can talk about that more later. But neurodegenerative disorders are a large and largely untreated population: Huntington's disease, Alzheimer's, Parkinson's, ALS, and others. Psychiatric diseases, schizophrenia, depression, anxiety, and what have you. There have been a lot of drugs that came out of the initial findings in the 1950s with chlorpromazine and others, that you could effectively treat some of the symptoms of things like schizophrenia in a percentage of the patients. But unfortunately, the field had not moved much away from the initial targets, and much of the work had been Me Too efforts from that point on, with both schizophrenia and depression. Infectious diseases are particularly nasty in the CNS because it's so hard to get the drugs in without significant side effects in to take care of infections, fungal diseases, bacterial infections, and like. There's specific sequelae from CNS injury, for example, a traumatic brain injury and spinal trauma, none of which have been treated very well: ischemic and hemorrhagic stroke, which I'll talk about in more detail, and epilepsies. Epilepsies, there are a lot of drugs that treat a percentage of epilepsy patients well, and more coming down. So it's one of those areas where there's a pretty decent level of success in treating. Pain syndromes, including migraine. There have been some recent advances with migraine, but in terms of general pain, opiates and their derivatives remain the most effective compounds, and most of the search for alternatives has failed over the last several decades. We have some new targets now that we are pursuing, but it's a very difficult area as well. And brain cancers, brain-specific cancers are some of the most tragic and fatal. Glioblastomas, for example, and that's actually a venture that I'm involved in, is trying to approach it with a new direction. And addiction. And again, this is only a fractional list of the things that affect the central nervous system. And I think it's sobering but true that if you live long enough, you'll have something that falls into one of those categories. And for most of them, particularly neurodegenerative disorders, there's currently very little we can do to ameliorate either the symptoms or the disease itself. The failure rates for many CNS targets have been extremely high. If you look at brain cancers, nearly 100%. I mean, there are some treatments, but they have very little extension in terms of how long you're going to live after diagnosis. For disease-modifying treatments of chronic neurodegenerative disorders, the failure rate's been nearly 100%, with the exception of multiple sclerosis. There have been a number of treatments at least for relapsing MS, and that's been one of the bright spots. Alzheimer's disease, obviously, I think it was just raised earlier. Aduhelm was approved recently, and it is an A-beta antibody treatment. It really is not very effective. There's some question as to whether it should ever have been approved. And this is actually part of the history of Alzheimer's disease. A number of things, even the palliative treatments, there's some question about their effectiveness. And was a large political pressure put on the FDA to get something out there because there was nothing. And so any glimmer of success is likely to be translated into approval that wouldn't have been granted in any other area. The patient advocacy groups have been very key in that process, and one can hardly blame them, for sure, in areas where there's been absolutely no success. But Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, ALS, there's been very little success. All of them have targets that look promising, and so we hope that there will be progress in the future, but it's been very slow going, very tough. Stroke, with the exception of the clot dissolving or removal agents such as TPA in the case of acute thromboembolic stroke and traumatic brain injury, have all been very, very tough. And I worked in the stroke area for quite a while. I will give you a, whoops, sorry. For some reason, my computer just went crazy. So why did this happen? Neuroscience came of age in the decades between the 1970s and the 1990s. Prior to that, it was really a very spotty discipline. But during that period, there was a huge explosion of knowledge in the area of neuroscience. We generated a whole bunch of hypotheses, and drug discovery programs were initiated based on a hypothesis, which is exactly what should have been done. However, we often thought, and I say we in a sort of royal sense, often thought we knew more than we did, which is clearly often leads down wrong paths. We often over-relied on academic experts. In other words, people would be brought in and they had a particular target that they thought was of great interest and would, basically accept little else but that in terms of a direction. I've personally been involved in programs that were completely driven by an academic finding, which turned out later to be either partial or even wrong. It would be fine as long as everyone agreed to go where the drug discovery data indicated. In other words, as we learn more about the disease, and often that knowledge comes with the actual program as it advances. If there was a feedback loop in fine tuning the direction of the program as a function of the data, that would be great, but that's often not the case. Hypotheses often become political, and a large amount of time and money is spent defending hypotheses rather than thinking deeply and freely about alternatives. That's the sunk cost fallacy, obviously. It is very, very frequently encountered in this area. One of the areas that I think has suffered from that is Alzheimer's disease. And, I would not say that the A-beta hypothesis, which has driven almost all of the work, or the tau hypothesis in terms of the targets, is wrong. But I think that the evidence has shown that they are not sufficient to altering the trajectory of those proteins. Mishandling within the CNS does not appear to be sufficient to greatly affect the outcome of the disease. And there are alternative hypotheses, and have been for many years, that the politics around the money involved in academic programs as well as in drug discovery have made it very difficult to pursue. And I think that has been to everyone's detriment. Eventual failures, which should be informative, often didn't lead to retooling of hypotheses, but rather to program rejection and wholesale abandonment of certain diseases. An example of that is ischemic stroke, where about $2 billion were spent in the late 90s and early 2000s, pursuing a number of ways of trying to block excitotoxicity associated with the stroke. All of them failed ultimately. And it was very, very difficult to get any company interested in picking up and going after acute stroke again. That started to change again in I think, a good way, but it took a long time. There was a very high activation energy barrier against doing that again. I think particularly difficult is animal models in the area of CNS drug discovery. Even in areas where there's seemingly good face validity, where the animal model of whatever the condition is looks like the human disease. These things were often constructed for convenience, and the result was very little predictive validity in terms of predicting the outcome of a drug effect on. Yeah, we could cure something in a rat model and take it into the human, and it did little to nothing. Marketing often dictated what science we could do rather than the other way around. I'll give you a concrete example of that in a bit. Regulatory agencies also took a very dim view until quite recently, actually, of multi-drug approaches to many CNS diseases in areas where no single drug had shown approvable efficacy. And you can understand their reticence about that. They're usually fine with cocktail drugs as long as the safety and efficacy of each component of the cocktail has been independently shown. And then you add them together, study the safety and efficacy of the cocktail, and on you go. But in the case of something like Alzheimer's disease, and I had meetings with them about this, and actually wrote with the former CNS head of the FDA and many other people a paper on new approaches that involve multi-drug therapies. So they are changing their opinion of that, but they were very, very hard to convince that when you make a cocktail out of a number of things that have shown no efficacy, that you may have emergent efficacy out of the cocktail that you don't see with any of the components. And you can understand their reticence, but I think that they are starting to change in their opinion, as long as they're safe. So I've talked several times about the example of ischemic stroke. About 87% of all strokes, about 800,000 per year in the US at least, are ischemic strokes, meaning that it isn't caused by a leakage of blood, a bleeding in the brain, but is instead caused by a blockage of the blood flow into a particular area. It's the fifth leading cause of death in the US, about 140,000 deaths per year in this country. Caused by a blood clot or an embolism, more often by a blood clot, I think. About 7 million survivors with varying degrees of disability, and it is the leading cause of long-term disability in the US. There have been really dramatic improvements in emergency and post-stroke care and support, and one of the things is getting people into the emergency room, making them aware of the fact that they may be having a stroke and seeking help earlier. And there was a whole campaign, the Time is Brain campaign in this country, both for the on the physician side and on the patient side, to try to get everything mobilized and people into an emergency care situation within this sort of narrow window when things can be minimized, at least the disability can be minimized. Clot reduction dissolution remains the most effective intervention, but there's always been a limited time window for treatment. It does not cure all of the aspects of that stroke in most cases. If you get in very early, it may, but in most cases, there is a reduction in the disability that results from the stroke if you get in early enough and get a clot reducer like TPA and get rid of the clot. So the thing that everyone operated on, including myself, when I was in charge of the stroke program, the ischemic strokes consist of a core region of tissue that most people, particularly then, considered to be unsalvageable. So it's a region in which, even if you recanalize, in other words, if you can take the clot away and recanalize, it is unlikely to ever be salvaged. And in these pictures, you can see the red area represents the core infarct. and then all of them have a penumbra of tissue at risk around them. And that penumbra is the area that all of us thought, which, by the way, should it die, should that area at risk be transformed into an area of death that's equivalent to the core infarct, the amount of disability or central nervous system dysfunction increases dramatically depending on where it is. And so the target of most stroke therapeutic programs was to reduce the effect of the stroke on that green area in these pictures, so that you could restrict the eventual dysfunction to those things that are really a function of that red area. The rationale for why we thought this would work is that those neurons in the green area largely still have some blood flow to them. And it was really a function of things that occurred in the red area that were affecting those neurons in the green areas that were causing the stroke, the infarct size to increase over time. So there's a massive release of neurotransmitter, both within the core infarct area, but also in neurons that are connected to it, both within the immediate penumbral area, but also even at some distal areas. The pathological levels of calcium entry and intracellular calcium release, and these initiate a whole group of processes that lead to cell death. And this was the excitatory cascade or excitotoxic cascade hypothesis. And over a period of about a decade, a number of mechanisms were postulated that could protect neurons at risk from this cascade. And the idea was that if you intervened within a certain period of time with either particular receptor antagonists, calcium channel blockers, in my case, potassium channel openers, caspase inhibitors, a whole laundry list of things, that you should be able to reduce the size of that penumbra and thereby reduce the infarct size. Many of them look good in animal models used, many were tested in the clinic, and every single one of them failed to demonstrate any significant efficacy after $2 to $3 billion were spent in clinical trials. I personally spent, my trials spent, I think, $156 million, and you can imagine when they didn't work that I was not feeling very secure in my position. And to the credit of Bristol Myers Squibb, whom I worked for at the time, they literally patted me on the back and said, "Well, that's what we do, Val. We test hypotheses, and by golly, this one didn't work. So go back to your lab and think of something else." Almost all efforts at neuroprotection were ended. They've started to limp back as we've understood more about the disease. But I think, and this is key, at least personally, I don't think that any of these hypotheses were adequately tested. They may not be valid. We don't know at this point. I just don't think they were tested properly. And I think that that's an important part of the history of this particular target that needs to be re-examined. We got almost everything wrong, except the need to initiate treatment quickly. And that was actually a very good thing. Has value even for the non-excitotoxic cascade treatments. People have studied very closely the treatment window for clot dissolving drugs and have been able to extend it very significantly. While the mechanisms of ischemic cell death and neuroprotection probably are valid, we clearly didn't understand the disease target. And one of the things that we didn't understand was how long that process continues. And so almost all of the treatments were designed to go after something very quickly and to have a very short period of treatment time. And in fact, and I will say this from the clinical trials that I was involved with, but it was very similar everywhere. Marketing people told us that we couldn't get reimbursed if the people were in the hospital, if this was given for more than 3 days. And so, Was there a rationale for doing that? Yes, but we now know that in fact the period of sensitivity extends far beyond 3 days. And that wasn't entirely known then. In fact, one of the outcomes of the trial that I was associated with really demonstrated part of that wasn't understood. Stroke evolves over a much longer time course, and treatment almost certainly has to be continued during the entire period of potential risk. So even some of these failed therapeutics may very well have worked. Instead of 3 days of treatment or 2 days of treatment, we now know that the strokes evolve over weeks, possibly even months. In other words, those neurons are still at risk for that entire period. And nobody tried putting that on for periods of weeks. Other aspects of the natural history of the disease were ignored or underappreciated, roles of inflammation, what have you. And in some cases, the ability of the therapeutic to cross the blood-brain barrier was completely ignored. There was one case that I won't mention the company, where we and everybody else had looked at the compound and knew that it couldn't get into the brain. And when we talked to people at meetings from that company and said, you guys are about to fire up what turned out to be a $200 million program or so to look at it in the clinic, it doesn't get into the brain. And they said, well, we're not worried about that. We think that the blood-brain barrier, sort of is disrupted in the area of the infarct. And, we all said, go with God, but it failed miserably. We don't know that drug wouldn't have worked had it been a compound that actually got into the brain and was kept there long enough. We didn't restrict who came into the clinical trials, I think, carefully enough. I mean, in my opinion, it's better to find cohorts for clinical study that are more similar, so that you can understand the outcome of the treatments better within a smaller group. I was involved as a chief scientific officer of a company that took a ALS drug into the clinic. It looked very good in phase 2, where we had an efficacy read. Pardon me. And in fact, so good, it was the best phase 2 trial that had ever been run in ALS. I was able to publish the results in Nature Medicine. Very tough to do. And it failed completely in phase 3 when it got out into a patient population that was, I think, much more heterogeneous than that had been used in the phase 2. And I think that that's often the case with neurodegenerative disorders in particular. Not enough attention was paid to the dosage form. Often the dosage form restricted the duration and amount of the drugs that could be given, often because of the lack of solubility of the drugs themselves. So they had to use excipients that weren't tolerated well. And again, drug cocktails in stroke, I think, are likely to be much more successful, and that was largely ignored at the time. And I think key is that animal models that used to propel drugs into the clinic were badly designed. And frankly, at least very quickly in the game, I knew it, and I think everybody did. We used, smooth cortical species such as rats and mice because of their, fast generation times and their cost. And they have very complicated central nervous systems, but they are not primates. They are not humans. Generally, the duration of the treatment was very short, and damage assessed after only a few days to a week. I won't go into great detail, but there were people that looked at it at longer and showed that any improvement that you saw at a week after a day of treatment was gone 2 weeks later, and nobody wanted to hear it. Young adult animals were used almost exclusively, at least during those early years. And most human strokes occur in older patients, and older patients have much lower levels of neuronal plasticity, ability to repair damage. Almost everything we did was too permissive. And clinical trials were designed to fit marketing priorities more than the science as the science evolved. And there are lots of other CNS disease areas where we could learn from our past mistakes. Alzheimer's disease is a clear example, a huge unmet medical need. Many failures, no real success in terms of disease modifying treatments. The current treatments are almost all palliatives, such as the acetylcholinesterase inhibitors, and they produce a temporary enhancement or marginal temporary cognition enhancement, but they don't really change the trajectory of the disease. Same thing is true of amyotrophic lateral sclerosis. Riluzole is the only successful drug. There are a couple others that are coming down the pike, but that are going to end up being very similar. It extends life by, I think, optimistically by several months. It doesn't really change the trajectory. It just sort of bumps it up. Parkinson's disease, largely palliative treatments again. Dopamimetics that will improve the tremor and what have you, but they don't really change the ultimate outcome of the disease. With pain, opiates and NSAIDs remain most effective treatments. Opiates, obviously, with lots of other side effects and issues with those, but they still remain some of the most effective pain drugs, despite the fact that we know how terrible they can be because nothing else has come in to take their place. And in psychiatry, most psychiatric drugs are ligands for biogenic amine G protein coupled receptors, following on very, decades old discoveries, accidental discoveries largely. They don't exploit new knowledge of other transmitter systems involved in psychiatric diseases. That is an area where that is changing. New drugs are in the pipeline that will be trying to exploit new knowledge in terms of transmitter systems other than biogenic amines. So effective ligands, I mean, the good news is that we can make effective ligands for almost any target. And that now includes small peptides and other large molecules that can get into the brain. So even though it's still difficult to pull a molecule up, show that it has the proper pharmacology and that it can get into the brain, it can be done. I'm not a medicinal chemist, so I'm not tooting my own horn, but medicinal chemists are impressive people. They can make things, they're sort of like engineers. As long as the science is there, they can eventually make something that works. That isn't the real barrier. The real barrier is making sure that we understand what the target does in disease and how that affects all of the other targets downstream from that. Understanding of disease processes has greatly improved. And in particular, the understanding of mutations leading to CNS disease has advanced meteorically. Although that doesn't always mean that it leaves you scratching your head more than it does necessarily finding a path to success. An example of that is schizophrenia, where there are lots and lots of mutations that seem to lead to the same sort of phenotypes, and it's going to take large databases and a lot of help to figure out which of those are the most important and how those can be targeted in ways that minimize the side effect profiles of the drugs. I think companies have changed in their approach to these targets as well. That it's better to have a successful treatment and learn how to market it. Something that there was huge resistance to in the 90s and 2000s. Than to try and force fit the discovery and development of a treatment to an existing market, standards of care, and current reimbursement limitations, which really affected, I think, the stroke therapeutic area. The FDA is more and more willing to entertain the idea of cocktail treatments, including cocktail treatments where none of the components necessarily would meet approvable levels of demonstrated efficacy. As long as everything is safe when put together, I think that they are much more likely to approve that than they would have, say, 10 years ago. there's renewed interest in many CNS targets, something that there was a period, sort of the valley of death in the late 2000s through the early 2010s. And many CNS targets, if you approached either funding agencies or VCs at the small company level, or went to executives in charge of therapeutic areas at the large company level, they would look at you and just smile, and you would get nowhere with them. And that has started to change in a great way. It is no longer off the table. For example, and a classic example of that is the single mutations in ion channels that lead to these fairly rare epilepsies. There has been a lot of support to go after those, and one of the ways that patient groups or the parents of folks who have these children with these diseases have been very instrumental in both getting the money and getting the political might behind advancing these things, even if they smack of personalized medicine and very, very, very orphan indications. There has been increasing support. And those of us at the bench level and clinicians at the bedside, and even the administrators of commercial entities, all remain committed to finding new treatments for CNS diseases and conditions. And I think that that bodes well for the future of CNS drug discovery and development. And I'd like to acknowledge the contributions of my colleague Leonard Kishmarek in pharmacology at Yale, and the hundreds of colleagues that I've worked with over the years. Thank you very much.
Thank you very much, Valentin. If you can close the slideshow, yeah, and we can go back to the floor. Depressing and gloomy story in terms of where we are with CNS drug discovery and the clinical successes we have so far. Interestingly, we talk a lot about cancer in these events, and this is the first time we got into CNS. However, some of us have exposure to this field, and cancer, it seems to be lucky here in terms of how many successes and how many cancers we've had. And ironically, the population seems to be a lot more affected by CNS in general than by cancer. So I see a question from Jack. I have some questions for you as well. So the floor is open for your participation. So Jack, please go ahead.
So first of all, I'd like to say thank you very much. I really, really enjoyed your talk. I'm interested in screening disease models and why they don't work. And I've read some of the literature, but it was very interesting hearing you talk about it firsthand. So thank you very much. And I guess I've got two questions. One is, why do you think there was insufficient sort of critical faculties applied to the models and what they might tell us? And do you think that's changing? And then secondly, well, no, let's do that one first.
So I think that part of it was that there was sort of a herd mentality. Everybody was doing this. It came out of academia. These models came out of academia. Some of us pushed back on them. I didn't like the idea of using rats. I literally had administrators say, yeah, but you know what it costs to use a monkey? And you're going to have to use 200 or 300 of them in a drug discovery program. And they cost almost as much as a human clinical trial to administer a drug to a monkey. Not happening. So go back and retool. And, I stood in front of a group that was a consortium of folks that were clinical trialists in stroke and who also had preclinical laboratories. And, they were, this is late in the game, probably 2003 or 2004, when we all recognized that the models weren't very good. By the way, all of the people in that thing, about 100 of them, had contributed in the literature using those models. So it wasn't as if they were standing outside of this. And they were also, oh, we're using the wrong models, they're too permissive, we're using this or these. I said, I'm not so sure that that's true, particularly with respect to the smooth cortex models for stroke. I just think that if you took old animals that represent the target population in humans and started doing some of these things and left the treatment on board longer, you would understand a great deal more about how these drugs work. And I got the pushback again that it's very slow to create old animals because 90% of them die before they get to that age. The cost of an old animal starts to approach that of a primate. So economics was a large driver of all of these problems of the models, as well as foolishness and foolishness on my part too. I'll freely admit it. I mean, we were all very naive and very hopeful, because we saw these dramatic effects in these models, and we thought that would translate immediately. And I'm sure in cancer that's true as well. I deal in brain cancers, and people have said, well, you can cure any cancer in a mouse, but so what? When that same thing goes into a human, nothing. So I think the cynicism about various rodent models in particular exists in many different areas.
Can I ask a second question, which is about, there's some interesting stuff going on about reclassifying psychiatric disease, i.e. looking for sort of underlying pathobiology rather than necessarily the diagnostic categories that the FDA has traditionally recognized. So for example, anhedonia would be something that you see in depression, you see it in schizophrenia, possibly other things as well. There are some agents which may, if anhedonia is indeed a sort of coherent pathological or pathobiological entity, some agents may treat it. But then you're stuck with the problem that the regulator still recognizes a different sort of disease classification. Is that something that you think is problematic, or is that something that's starting to change?
Well, I think that what that would probably end up forcing people into doing, and I've thought a lot about psychiatric disease recently. I agree that treating particular symptomatology is probably going to be fruitful. But for example, going after, when you look at, for example, schizophrenia, the negative systems include many things like anhedonia, as opposed to, direct fragmented thought. And I think that you're going to have to put a cocktail together that treats a number of these things in order to get a disease modality effectively treated. And it may be a different cocktail for depression than it is, or for bipolar, the manic phase of bipolar disease, than it is for psychiatry. But I agree with you. I think that that's actually a very promising way of looking at it. But as you know, trying to turn that Titanic, on a dime is very, very tough. It takes a long time to turn a big boat, like the thinking about an area that's evolved over 100 years. But I do think that's fruitful. I do think it's what's going to actually be successful.
Well, thank you very much for that, and also thank you very much for the talk, which I really enjoyed.
Thank you.
Thank you, Jack. John, please go ahead.
Yeah, thank you, Valentin. Len wasn't making a cup of tea, by the way. They couldn't sign in, so he sent apologies. There's a few people who wanted to watch, and they've struggled to sign in. I don't know why. Anyway, thank you for a great talk. I mean, the parallels, as Lisa said, with cancer are really amazing. And I think in the field of oncology, which you also know a bit about, people are saying that it's reaching a sort of plateau phase in terms of treatment, and that we should be looking at alternative ways of looking at, how would you call it, health and cancer. So prevention, early diagnosis. So if CNS is also on a plateau with some sort of nice little bumps happening every so often, what's the alternative there?
Well, I think that it's a little bit different. I mean, clearly, I think cellular mechanisms of cancer, while not completely understood, are understood a bit better. The tumors tend to be a bit more uniform, and I think that there isn't so much a plateau with CNS as there was just a period of, being lost in the Sargasso Sea, and if we can get a wind to come up, we can actually get out of that, because it was lack of understanding, or at least a need to further advance our understanding of each of the diseases, that has driven new interest and led to the discovery of new targets. I just think that we had an inadequate understanding of our targets going forward. I do agree with you, for example, that and I think everyone in the field would, that looking at these diseases earlier on, trying to find biomarkers, for example, which don't exist in most CNS diseases, so that not only can you predict the effect of drugs, but you can start to identify patients before they have a frank expression of the symptoms, at which point it may not be possible to reverse it, particularly with the CNS. Is critical to development of effective therapies. We are at sort of a plateau, particularly with psychiatry. We've been at a plateau for a long time. I mean, they've been, basically what's happened in the industry for a long time is that they just rearrange ligands for the same subset of receptors over and over again, and they get a little bit of an improvement in one thing, maybe a little bit less tardive dyskinesia associated with a schizophrenia drug, maybe not, but they've never made major inroads, for example, into what are called the negative symptoms or cognitive difficulties associated with schizophrenia. And it's all because they were locked within a certain subscribed group of targets. But now they're starting to get outside of that. There are many, many more targets to exploit. And as long as the money and the will is there, I think eventually they will make some big inroads. I don't know if that really answers your question, but
yeah, I think so. I just say that you're quite right. I mean, the understanding of cancer has been explosive over the last 30 years, say. And I think what it's done is to tell us that the problem is so complicated that it's unlikely that we're able to affect it by pharmacological means. Yeah. And so, the knowledge is there, but the biology tells us that the complexity is a barrier to what we want to do. And I wondered if that's the same in the
Well, I do think there are parallels there. I mean, people have said that the central nervous system of a human is the most complicated thing in the universe, and involves more synaptic interactions than there are discrete particles of matter in the universe, which is anyway, there are a lot of them, and there are a lot of interactions, and we've only scratched the surface at understanding them. So yeah, I mean, it is extremely complicated. Behavior is extremely complicated, and all behavior is a function of the central nervous system. So it's very, very, very difficult. Overlaid with that are the effects of any drugs on behavior, and trying to avoid, the do no harm thing is very difficult to avoid with CNS. And it is unbelievably complex, and more complex the more we know about it. It is clearly going to take a lot of computing power to try to start to see paths that are worthwhile going down while avoiding the things that we shouldn't go down, and that's only now starting to be done, I think, on a logical scale.
Thank you. I think one of the key differences between cancers and CNS is that with cancer, we have amazing solutions in terms of surgery, radiology, and some magic bullets like imatinib, like a few other drugs which made amazing difference. And there's a lot of conversation of cancer becoming a chronic disease, and I think it's a kind of virtuous cycle where more investment keeps going on, and this kind of failures in cancer tons of them, they become slightly less noticeable because at least we have some successes in some cancers. With CNS, we don't seem to have that kind of encouragement from many sides, and looks like for some diseases, there's hope for those that have genetic origin, but they, so far as we know, it seems to be a small percentage. And maybe those have a chance of disease modifying therapies, but vast majority of them so far will probably lie down still along this path of trying to get kind of pharmacological interventions. Do you see any other path for CNS beyond pharmacological interventions? What else can be tried? Or the real solution, just like you suggested, is actually going back to the drawing board, trying to understand the mechanism of the disease and keep trying down the pharmacological path?
Well, I mean, I think, I mean, if somebody I wish that we could prevent diseases by understanding what in someone's lifestyle or what have you would contribute to eventual expression of a disease phenotype. And I think that to the degree that that's applicable, that should be done. I think that any intervention that can help to prevent disease is clearly better than something that tries to cure it. But many of these diseases are a function of aging, and as coupled with lifestyle as well as genetic components. And so some of those I think we can tweak, but ultimately, and this is my business, so this is my approach, obviously, you're going to have to find some way of intervening in that disease process, and that turns out to be pharmacological, however you define that. I mean, the definition of what's pharmacological, used to be nothing but fungal alkaloids going to small, synthetic small molecules. Now it's peptides, antisense nucleotides, and what have you. So what constitutes a pharmacological intervention is going to continue to change and evolve over time. It won't be the sort of hammer necessarily that it was in the past. But I have high hopes for cocktail treatments for some of these more complicated diseases. I think that the fellow that brought up the idea of going after particular symptom modalities in psychiatry is actually, I think, a brilliant way of sort of going after something that a number of us have been trying to direct people towards. But politics is a huge issue in this area. And anytime there's money involved, politics is sure to follow. And I've had a long career of sparring. Anybody who actually knows me in the profession knows that I have many times, been shown the door by those with more money and political strength than I had. But I have confronted them for decades. And sometimes it's worked, and slowly but surely, people have been brought around to a different way of thinking, whether it's correct or not, at least a different way of thinking and a different approach. I mean, I'll give you a concrete example. There's a lot of evidence that Alzheimer's disease is really a metabolic disease, and some people have started calling it, type 3 diabetes. That insulin resistance on the part of neurons. I mean, neurons use more energy than any other comparable cells, or almost any comparable cells in the body. And so, the development of a metabolic disease associated with neurons is going to express itself in some very dire ways. And there are increasing subgroups of people who believe that is the genesis of many of the sporadic forms of a large number of these neurodegenerative disorders. Now, that gets to your question, John, I think, about are there other ways that you can approach it? I mean, diet, exercise, and everything else contributes to metabolic disorder, or, lack thereof, or, the wrong diet. And so there are, if we know that as a certainty, then lifestyle changes could certainly contribute to a reduction in both the frequency and severity of some of these neurodegenerative disorders. I mean, Parkinson's disease is a classic example. If you're a boxer, you're almost certainly going to get Parkinson's disease. If you use your head to if you're in Manchester United and you're always doing headers in, you're more likely to end up with a neurodegenerative disease, either dementia or Parkinson's disease. Same thing with American football players, almost all of whom it's becoming obvious to us. When you have 300-pound people crashing headfirst into each other and doing it for years, the outcome of that is predictable. So we know that lifestyle changes contribute at least to some of those cohorts of patients. And so that is a non-pharmacological way understanding more about the disease. I mean, the whole thing with CTE, nobody believed it. Nobody wanted to believe it because there was money involved in American football, at least. And it took a lot of fairly brave people, I think, to speak truth to power and get that accepted as a major problem with certain sports and other types of lifestyles. So, I think that the more we understand, the more we can do both, get better pharmacological treatments and inform us about how lifestyle treatments can better or reduce the probability and better the outcome in those diseases.
Thank you. Any other questions from the audience?
Well, thank you very much for this opportunity. I enjoyed it.
Thank you, Valentin. Fantastic talk. We learned a lot from you. So we will have recording available tomorrow. So definitely feel free to send the links to people who missed the talk. And we wish you success on behalf of all of us who may suffer from CNS or our relatives. So thank you so much. Bye-bye, everyone. We'll see you in 2 weeks.
Bye-bye.